mechanism is decide which exon to skip? So this is a very interesting question. Till nowadays, scientists still working on that, and they have provide several different mechanisms. But one of the mechanisms that have been agreed recently, been proved by experiment, is when the splicing sum come down and trying to remove the intron. So some intron will have a variant or mutation which is in the location nearby of the uh, which is the region nearby to the exon. So this is intron, and this is the region located in the intron, but located nearby the exon in here. So the variant, the DNA sequence in the intron in here might have an effect on the splicing sum and decide of those which exon to putting in the mature mRNA and which exon will be skipped during splicing process. Okay, and there is more variants. There are more mechanisms that you can find on this paper. Okay, so I will put the link in here to you. So this is a very recent research published on 2015. Sorry, so this is a very interesting research published recently in 2015 on Biomed report. We study about the mechanism of alternative splicing, just released like 7 years ago, and they have provided several hypotheses how the splicing process was put in. So the three main possible mechanisms: exon shuffling, exonization, and consistently splicing exon. And all the information about research they have provided in the paper here. Okay. All right. So come back to our lesson. Any other question? All right. So three main components that played a role in the translations. First is the mRNA, which used as a template. So this is the template. For the translation. So the ribosomes, which is composed by two units, the last one and the small one, is play a role as a machine to synthesize the polypeptide. And the last one, tRNA here, is play a role as an ingredient. Okay. So the tRNA here is have a safe of the pre-leaf grass, and one in one direction is have an anticodon is the trinucleotide that binding into the template here, and in one end it's attached to amino acid. The ribosome, which play role as machine to synthesize the polypeptide here, have two subunits. We call it a large subunit and a small subunit. But a very, very recent publication published right in 2019 say that small EBIC in Arabidopsis mitochondrial ribosome. That means in Arabidopsis, a plant that was used as a model for many genetic research in plant, the small subunits even bigger than the large subunit. So we call it the large small subunit, but it had the size, the 3D structure size, even bigger than the large one. Okay. So in some species it's like that. So the tRNA here is have a safe three-leaf rust, and in one it's attached to the amino acid, and in here is the anticodon. Okay, which is coding for the tRNA. So this is the genetic, the universal genetic code that have us to translate from the information of mRNA to the language of polypeptide. So, for example, if we have a codon called UAU, so how to use it? So we go from the first one here, A. So we come here and U. So that means it's encoding for tyrosine. Okay. And so, for example, GCA, let's GCA by the same way, come from the first letter here, G, and next one is C, so we go to C, and the last one is A, so is encoding for alanine. So GCA is coding for alanine. Okay. So the translation process start when a small subunit combined with a large subunit of ribosome and combined to the mRNA as a template. So we have the mRNA here. But the translation will not start right at the position of the attached site. It will slide until it reaches the start codon. So AUG here, this is the start codon. So that means the position here to the beginning is the 5' UTR, right? 5' untranslated region. So as soon as it's reached the start codon, so the tRNA carrying the start amino acids, methionine, will come and binding into the sequence. So the tRNA carrying methionine will have the anticodon U, A, and C, which is complementary sequence for AUG, the start codon. So in the ribosome, there's two positions. The first one here, and the second one. Next, the next tRNA will come and binding and release the amino acid here. The catalytic site will enhance a polypeptide link. Polypeptide is actually a covalent link, so it's really, really stable between methionine, the first amino acid, and the second amino acid one. And then it the methionine form a peptide bond in here, and the tRNA that carry methionine was flushed out of the position, the first position, and the tRNA here was transferred from the second position to the first position. Yeah. Okay, and another tRNA come and fill in the second position, and so on, so on. We have the tRNA. So the peptide bond in here from amino acid, we will have the alpha carbon, which is the carbon in the middle here, binding to the side group, and we have called it amino acid because it has an amine group. And the carboxyl group here, or we call it the acid group. So the peptide bond will form from the amine group of the second polypeptide, second amino acid, with the first with the carboxyl group of the first amino acid, and form one water and one peptide bond. And as I say, this is a covalent link, so it's really stable. And meanwhile, in DNA, we have the 5' and 3' direction. In amino acid, we have also, okay, wait for me, I open my paint file. Open a new one. So, excuse me, can you turn off your microphone? Can you mute yourself, please? Thank you. So, meanwhile, in DNA, we have the 5' direction and 3' direction in DNA. Then in the polypeptide, we have also the amine group, or the N terminal, and the C terminal, carbon terminal. Of the amino acid sequence. Okay. And the translation process finished, and since the ribosome reached the stop codon. So we have 3 stop codon: is UAG, UGA, and UAA. Since it reached the stop codon, it's when release the polypeptide and finish the translation here. And the polypeptide will directly go to the post translation modification to form the 3D structure of protein. So let's watch a small clip to demonstrate how translation process. So this is the nucleus, and this is nucleus membrane, and you can see here the yellow region in here is the hole in the nucleus membrane. So we have the mRNA, the mature mRNA here have been finished the maturing process inside the nucleus membrane, and it go into the cytoplasm through the hole in the nucleus membrane. Okay, to start the translation. Oh. So we have ribosome, the small subunit, and the large subunit come and binding into the mRNA to start the translation. So we have the floating in here is the tRNA. The tRNA carrying the suitable amino acid. And we have the product is a polypeptide, which is marked in the red string in here is a polypeptide. So you can see here each tRNA come and gives the ribosome will make a peptide bond between the amino acid of the polypeptide chain here with a new amino acid that the tRNA bring along. Okay. So we see that a tRNA here, which is have a red dot in here, is this red dot is the amino acid that the tRNA carry to the ribosome position. Synthesize the protein peptide. So different type of amino acid. Oh. Different type of amino acids. So the polypeptide here. So as soon as the polypeptide is being synthesized, it start to through post-translation modification to form its 3D shape of the protein. So as soon as the protein, as soon as the polypeptide was being synthesized, it had been through the post-translation modification to form its higher structure. So the process called protein folding. So there are four main levels of the structure of protein. So the first level, the primary level, is the sequence of the amino acids. So here, this is the first degree of proteins, first level of protein structure. So the second one is a loop here. For example, the formed by the hydrogen bond from one amino acid to the other, the hydrogen bond, we make to form the alpha helix, which is structure helix here, or sometimes it calls from another structure called the beta helix. Sorry, beta sheet. It's like a sheet of paper from here. So this structure, the secondary structure, the alpha helix or beta sheet, will combine together by several bonds that not covalent bond. This covalent bond could be the hydrogen bond or ionic bond, disulfide bond, one-f bond, or hydrophobic bond, to form a third structure here with several structure of alpha helix and sometimes beta sheet to form a third structure. And in some protein, in several protein, some subunit even combine together to form a more complex structure called the quaternary structure protein. For example, in the hematology here, we have three ternary structure of hematology: 1, 2, 3, 4. We have 4 subunit of hematology combined together to form a hemoglobin with the HAM. That means the iron atom inside. So let's watch a small demonstration about the structure, different structure of protein, and how protein was maintained its function due on the structure of it.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons. Learn more about the functions and 3D structures of proteins and other molecular machines at the RCSB Protein Data Bank.
So this is a very interesting data bank. Beside of the genome data bank is the protein data bank. So if you go into protein data bank, you type the three letters coding here, it will lead into the 3D structure of the protein in here. For example, I type 3W14 in the third panel of the protein data bank. It will provide me the 3D structure of insulin. So let's take, for example, I'm going to the protein data bank. Here, RCSB page. And I type D3314 Y14. So insulin receptor. So this is the 3D structure, a reform structure of the we can see it in 3D structure here. Click on this, wait a few seconds. It's really slow because it contains a huge amount of data. So wait a few seconds more. Here you can export. So here is the label ribbon of. You can pin it in the 3D structure to explore it, to investigate if how it was happening. So I even can zoom in different type of it. Okay. Anyway, it's a very interesting tool. Okay, so the different structure level in here. So the primary structure is formed by the peptide link, which is a covalent link, and the covalent link is really stable to temperature. And but the higher structure, the secondary structure, technology, is formed on the non-covalent link. That's why it's very sensitive with other factors that like temperature. Okay, so it's easy to use temperature to break the 3D structure of protein, because all the higher secondary and higher structure of protein was formed by the non-covalent link, like hydrogen bond or van der Waals bonds or hydrophobic bonds. So as a consequences from one sequence of amino acid can, based on the different post-translation modification, can form different structure of protein. So with one polypeptide sequence, we will have different isoform of protein, and different isoform will maintain different function. So as a result, from one DNA through a splicing process, will form different type of RNA. So we have different RNA variant of one DNA, one genes, and different RNA variant through the post-translation modification can form different type of protein. So as a result, the number of proteins will be much higher than the number of genes, always greater than the number of genes. Okay. So now to finish today's lecture, I have give you a exercise. So this is provide sequence of a genes with the full sequence that had categorized for you. So this is the promoter, this exon, intron, and exon. So it's a gene with 3 exon and 2 intron. Please identify for me where does the transcription start. What is the sequence of the mRNA precursor? That means the immature mRNA. Okay, sorry, immature mRNA. Follow the English pronunciation. The mature messenger RNA. Where does translation start, and what is the peptide sequence? Write down for me the first 10 residues of a peptide sequence, and where does the translation start? Okay, please help me to identify and answer those questions.
So is this the coding strand? Is this sequence the coding strand or the template strand?
So this is the coding strand.
Okay.
Yeah, so this coding strand from the 5' to 3' of the coding strand. Sorry. So for your convenience, I will copy the sequence here and put it in the chat box for you. Okay. Okay, so I have the first answer. Wait for me a minute. And
Excuse me, can you check back the file? Like, can you go back to the slide?
yeah, here.
There's a wrong here. There's something wrong here. Like the mature messenger RNA is wrong. I will take it back. That the two got replaced.
Okay.
So with you. Yeah, I love Python. Yo.
Okay. So the first question, where does transcription start? So we have identified it, studied in today's lecture, right? Those transcription start. In here, right after the promoter, right? So the transcription start in the position +1 here. Transcription starting site. Okay. So the transcription starting in the exon 1 in the position here. So this is the position +1, or the transcription starting site. Second question, what is the sequence of the messenger RNA precursor that the immature mRNA? What is the sequence of the mature messenger RNA? So we came back to the same slide. So this is the premature mRNA. So it including also all the exon and all the intron. And the mature mRNA is including only the exon. So as a result, we can see that the precursor is including all the sequence from here to here, and the mature messenger RNA is including only the exon. So exon 1, exon 2, and exon 3. Okay. All right. So your answer is correct. So where does translation start? Translation
starts on AUG or on the AUG.
Yeah. So we come from the transcription starting site. We scan in here until we reach the AUG or ATG on the sequence. So the first ATG.
I found the index is 68 or 69, like depending on if the string is 0 index or 1 index.
I'm sorry. What is the question?
I use Python to do it. Like I found an
okay.
ATG.
Yeah, but the first ATG, not the second one. Okay. What is the first
one? Yeah, the first
so,
one. The first one.
So in this position, right? Or in other? Okay.
Yeah. Position 68 on Python.
68. Okay, great. Okay. So you use Python to use it. So online we have several Python's pipeline to guide us to how to find out the translation starting site of a genes. So you can use it up if you like, not a problem. Okay. And what is the peptide sequence? Can you put it in the chat box for me? So we have ATGs coding for methionine. We have the next 3-1 here, next 3-1 here, here, here. So you can use the universal code. So for example, AAT. T is replaced by U. So it's asparagine. Okay. ASB, and so on. So on like this, you have 10 residual from it. And what does the translation stop? So this question is tricky. So you have to follow this ATG pre-nucleotide pre-1, 3 until you reach the stop codon. Okay. Remember that if you have a stop codon like here,
There is a UTC that ends at index
so here. Okay, so we have the stop codon is here. Okay, so that means that the translation will stop here. So that means all the region here is a 5' UTR, and all this region here and to the exon 3 is a 3' UTR. Okay. All right, so I guess that is the end of today's section. Do you have any question, comment? Okay, so since you have no more question or comment, I will finish the today's section here. Thank you very much for your attention and see you on Thursday.
Goodbye, Professor.
Goodbye, Professor.
Goodbye, Professor.
Goodbye.
Goodbye,
Thank
Professor.
you. Thank you. See you.
Goodbye.
So I received a question. Could we bring the genetic cheat sheet to the exam? What do you mean the genetic cheat sheet? And again, please send it to everyone. What do you mean the? No, you cannot bring that to the exam. Sorry, but for the, if you asking for this one, I will print this one into the question sheet for you, so you don't have to remember this. Okay. Okay. Is that answer your question? So you don't have to remember the genetic code, because I will print it directly into the question answers, the question sheet for you, so don't have to remember it.